Welcome back to Unit 3, Lesson 1, Part 3. Again, this part is optional unless you were away. We should have done it in class. Okay. So we're just going to pick up with the structure of RNA, which is a little bit simpler than DNA in the respect that DNA is typically double-stranded. RNA is typically single-stranded except for when it's being produced, in which case it's complementary binding to DNA. But that's only a temporary state. So we'll draw its key or main state. So we'll start with, just like DNA, you have to start with a nucleotide. But of course this time your sugar is going to be different. It's going to be ribose with a base attached. You can pick any base you want, but let's start with A. We'll show that it's ribose, but because it has that OH. And then the 3' carbon is going to attach to the next nucleotide. Pick another base, but be careful. Which one can't you pick? It's correct, no T's. But you could pick instead U. Attaches to the next nucleotide. Still building just like DNA, 5' to 3'. Pick another base, maybe G. And finally attaches to your last. And what would the last one be? That's right, C. And let's just go back and add in all the OHs to make it clear that we're talking ribose, not deoxyribose. And once again, just like in DNA, this structure grew 5' to 3'. But because there's only one side, we don't have to worry about that whole parallel-antiparallel discussion. And we don't have rails so much because there's only one side. Instead, we refer to it simply as the sugar phosphate backbone. So this section here would be the sugar phosphate backbone of RNA. Okay, and that's the structure of DNA and RNA. Now we want to look at the locations and functions. What do they actually do with these various structures? So the first thing is the birthplace. Where are these guys originally made? So DNA is made in cell nucleus. Likewise, RNA is made in cell nucleus. The workplace of DNA is the cell nucleus. In contrast, while RNA is still made in the nucleus, it works in the cell cytosol. That is the jelly stuff between the membrane and the nucleus, where a lot of the cell functions occur. Okay, so let's look specifically at the function of DNA. DNA gets a cool nickname. It's called the blank of life. Think about an architect. That's right. Blueprint of life. And it contains thousands, in fact hundreds of thousands, but thousands per chromosome, thousands of genes which are the blueprints for building proteins. And DNA is a very special blueprint because it's the original. So it's the original blueprint for the entire body. That must be protected. And copied to be passed from one cell or generation to the next. RNA there's several types of RNA, but RNA typically helps to convert DNA blueprint into an actual protein. So they're made in the nucleus, they work in the cytosol to build the protein. And there's three main types that you should recall from grade 11. The first being mRNA. What does that mean? The M. That's right. Messenger. And it is the working copy of a blueprint. Whoops. For a single protein. So we don't copy the entire DNA for every possible protein. When we only want to build one protein, we only copy the DNA for that little protein. And we'll talk more about that when we talk about transcription. And it is linear and single-stranded, unlike DNA, which is double-stranded. Linear in that it's not all twisted, it's a nice straight line, easy to read. Then we have tRNA. What's the T stand for? That's right. Transfer RNA. Helps transfer amino acids. Those are the monomers or building blocks of proteins. Amino acids to a growing peptide chain. Think back to last unit, peptide being multiple amino acids together according to mRNA code. So mRNA says which amino acid to put where, the tRNA actually brings it in. So one's the blueprint, one's more like the grunt worker on the construction site, picking up the amino acids and delivering it to where it's supposed to. And it is T-shaped, not why it's named that, just by fluke. And it's also single-stranded, which we're just going to abbreviate SS. Finally, the last one, do you guys remember? We have mRNA, tRNA, and good, rRNA, which equals, tough one, ribosomal RNA. Which constitutes a ribosome when partnered with a protein. So by constitutes, I mean if you have ribosomal RNA and you mix it with a protein, you now get a functional ribosome. Okay, so it constitutes a ribosome when partnered with a protein. The ribosome acts as an enzyme to form peptide bonds. It does a dehydration synthesis between the amino acids, forming peptide bonds between amino acids. So the mRNA tells you which amino acid to bring in, the transfer RNA actually brings it in, and then the ribosomal RNA, as part of a ribosome, actually links the amino acids together. So all three of them work together to build that protein based on the original DNA blueprint. Okay. We'll go into the details of how that's done, how we make copies of DNA and how we make RNA and how we build proteins over the next few lessons. However, if you have any questions on this lesson, please feel free to come see me in class. Otherwise, I'll see you in class, and I hope you have a good day. Thank you.